Hello, my name is Kyle Malter. I'm a fifth-year PhD student in the lab of Dr. Nicholas Shikuma. And I'm going to talk to you today about how bacterial injection systems stimulate animal development. So as you may know, bacteria evolved on this earth over 1 billion years ago, and animals not so long ago. So when animals have developed, they have developed in a world completely taken over by bacteria already. And so we know that bacteria influence animals and their development, but the major question our lab is interested in is how is this occurring? So how are bacteria influencing animals? So this is a coral, and corals are known to be require bacteria to undergo metamorphosis. And what we're curious in answering is how is that occurring? And so one of the things we've discovered is the way that some of these interactions are taking place is through contractile injection systems. And so what are contractile injection systems? So contractile injection systems are similar to bacteriophage. So I'm going to draw a phage. These are viruses that infect bacteria with their DNA. So this is a phage. And contractile injection systems are similar to phage, but they are not phage. So they have many of the machinery parts of a phage, such as the contractile tail and sheath and inner tube. But these usually inject, instead of injecting DNA, they inject a protein. And so one of the ways bacteria use these contractile injection systems are to inject toxins into target cells. And so what we've discovered is that there's another type of this contractile injection system that is released from the cell. So these are ECIS, extracellular contractile injection systems. And these extracellular contractile injection systems are required to stimulate a marine invertebrate's metamorphosis. So our lab studies a marine invertebrate. So we study a tube worm, H. elegans. This is the larvae. And in order to go from their larval stage into their adult stage, this is the adult tube worm, they actually require these ECIS. So in order for this to occur, you need the ECIS to take place. And what my project is interested in is how are these ECIS stimulating this metamorphosis? And so what we found is, let's zoom in here onto a singular cell from the Hydroides elegans. What we found is that these ECIS come sit on the membrane and using a tube and contractile sheath inject a protein effector. So these are the protein effector. Across the membrane. And I'm looking at the signaling systems involved with this protein effector. And so what we found is this protein effector then goes and sits on a membrane and stimulates an increase in diacylglycerol production, which is a lipid product from a phospholipid. And that diacylglycerol stimulates a known signaling pathway within the cell, PKC signaling. That then goes on to stimulate MAP kinase signaling. And from there, we see metamorphosis signaling. So more downstream signaling causing this metamorphosis to occur. And so what I found is that we see bacteria that are producing a extracellular contractile injection system that's then released from the cell. That extracellular contractile injection system is then finding the Hydroides elegans, injecting a protein effector across, and then stimulating this protein kinase signaling cascade through the increase of diacylglycerol and through MAP kinase to stimulate metamorphosis. And so what we want to do, what we want to follow up on next is understand this in a more broad context, looking at other animals such as corals, as well as we see these systems in human gut microbiota as well. And so we're interested in seeing how those are functioning. So thank you very much.